So today we're going to talk about action potentials. Now I had you watch a couple of videos, and now what I want to do is I want to take all of those complex processes that are happening in a neuron as it sends that electrical signal, and I want to boil it down to one drawing and kind of summarize everything that you watched. So the first thing that we need to do is draw a neuron. So we'll do a really terrible drawing of a neuron here. And we need to put some little projections at this end. So then, before we get started on the action potential, let's go ahead and just label the different parts of our neuron. So these structures sticking out of this end are the dendrites. This part that has the nucleus in it is the soma, or cell body. This part is my axon, and then these are my axon terminals. And my action potential is going to travel in this direction. So it will be received, the signal will be received by the dendrites, and then it will be transmitted along my axon and then out my axon terminals, where it will connect with either another neuron or another cell at a space called a synapse. Now, in the videos you learned that you can have myelinated axons, so we could draw Schwann cells, for example, around this axon and make this axon myelinated. But it gets kind of messy in the drawing, so for now let's just leave that off. And the next thing we need to look at are what's happening when my cell is at rest. So you'll notice there's a drawing that's like this and probably a bit more detailed and neater online that I've done. And it breaks this up into kind of three regions. So I kind of divide my cell up into 1, 2, 3 parts, but really everything is happening along this entire neuron. Before we can talk about what's happening when a neuron fires, we need to know how it stays ready to fire. So the inside of my neuron needs to have a resting membrane potential of minus 70 millivolts. What that means is the inside of my cell needs to be way more negative than the outside of my cell. In order to do that, we need to have a distribution of ions inside and outside so that my inside can be more negative. So when I'm at rest, my ECF, or my outside of my cell, is going to have more sodium ions and more chloride ions. And the inside of my cell is going to have more potassium ions and big negatively charged proteins. Now my proteins are too big to diffuse across the membrane, and my chloride ions can diffuse into where there's fewer chlorides, but they're going to do so very slowly. They're not really going to want to do that because the inside is already really negative. So those two negatives repel each other. So we're not going to worry so much about my chloride right now. My positive ions, however, my sodium and my potassium, can leak across the cell membrane. So the potassium leaks down its concentration gradient, so it leaves the cell to where there's less potassium. And my sodium leaks in to where there's less sodium. Okay, so it's going down its concentration gradient. Now, the sodium is going to leak in faster than my potassium leaks out because it is diffusing down its gradient and it is attracted to this negative resting membrane potential inside. So it's going to come in a little bit faster. If I were to just let these ions keep leaking, eventually enough sodium would leak in that my -70 would move upwards, become less negative. And if it hits kind of a magical number we call -55 or -60 millivolts, that's threshold, then my neuron is going to fire. So if the inside reaches -55, I'm going to fire. And it's going to do that by opening a bunch of gated channels that are going to allow ions just to flood into the cell. Now, we don't want our neurons to fire haphazardly. We want them only to fire when we need them to. So when my cell's at rest, I'm also going to need a little pump in my membrane here. And we'll call this the sodium-potassium pump. All right, so that's my sodium-potassium exchange pump. And if you think about a boat that has water leaking into it, back near the motor you may have what's called a bilge pump that helps to keep pumping water out of the boat back to where there's already more water. Well, my sodium-potassium exchange pump does something similar. It puts the sodium and the chloride back where they belong. So it's going to pump my sodiums out, and it's going to pump potassiums back in. It's going to pump the sodium out a little bit faster than the potassium in, kind of like how they leaked that way. So we're going to end up with 3 sodiums getting pumped out for every 2 potassiums that get pumped in. And that means that that pump is going to help us to maintain our resting membrane potential by putting the ions back where they came from. Now, if I want this neuron to fire, I'm going to stimulate my cell, probably out here at the dendrites, and I'm going to cause some little doors to open up called sodium gates. So let's draw kind of a little line here. Oops. So if the inside reaches -55 millivolts, then sodium gates are going to open. So if I stimulate my cell, I typically open up a few small sodium channels, and some sodium starts to leak in or diffuse in. If enough sodium diffuses in that the inside hits this threshold, -55, then these other sodium gates are going to open wide up. So imagine that I have a membrane here, and here's a protein channel, and it's closed. And then at -55, it opens. So what's going to happen? Where do I have more sodium? I have more sodium on the outside of my cell. So that means I have sodium out here. And if I open a bunch of doorways in my membrane that allow sodium to fit through, sodium is going to pour in. So sodium rushes in. And it's going to do that until the inside of my cell hits +30 millivolts. So it's going to kind of overshoot 0. Now, imagine that I've stimulated my cell down here at the dendrite, and I get an action potential. I get sodium rushing in. Sodium rushes in here, and then it diffuses down a little ways and makes this part of the membrane come to threshold. So then more sodium channels open, more sodium pours in and diffuses down, and now this part comes to threshold. More sodium gates open, more pours in, and then this part comes to threshold, and so on. So we get a wave traveling down this neuron. And that's basically your action potential. When I'm done firing, I need to reset that cell. So what's going to happen is, when the inside reaches positive 30, my sodium gates close. Because if I leave the sodium gates open, sodium just keeps pouring into the cell, and we don't want that. So what I'm going to do then is I'm going to open potassium gates. Now, where did we say there was more potassium? There was more potassium on the inside, right? So if we put a bunch of potassium in here, potassium is going to start to rush out. So potassium rushes out. Until the inside is back to its resting membrane potential. And that happens like a wave right behind my action potential. So here I've got sodium pouring in and moving down. That's my action potential. And then right behind it, I have potassium moving out and resetting that membrane as we head down. Now, if you remember back from chemistry, I said when you hear the word polar or polarity or polarization, I want you to think difference. So when I start off with the inside really negative compared to the outside, we say that the membrane is polarized. As sodium rushes in, I'm getting rid of that electrical difference. So I am depolarizing. So this is depolarization. So depolarization is basically my action potential. And then when I'm resetting that, I'm getting my difference back. So then this is repolarization. And then, of course, once we get to the synapse, we're going to have to do something there because we basically put a letter in the mail and it traveled all the way through the Postal Service and got to our family member's town, but it still has to get put in a mailbox before they're going to get the letter. All right, so let's look at it again real quick. We start off with my membrane polarized. The inside is very negative, and that's because of the balance between sodium and chloride outside and potassium and negative proteins inside. My positive ions are leaking across. So these guys leak. So my sodium-potassium exchange pump counters the leak, and that helps me to maintain this resting membrane potential. If I stimulate a neuron, some sodium moves in. If enough sodium moves in that I hit threshold, then sodium gates are going to open and sodium pours in all the way down this membrane. That is depolarization, and that is also basically my action potential. And then I'm going to close my sodium gates when I hit positive 30. I'm going to open up potassium gates, and potassium is going to pour out as it moves down that membrane like a wave. And that's going to reset my resting membrane potential. Now I can put all of this electrical activity into a graph, and who doesn't really love a good graph? All right. So if I look at this graph, this is over time, and this is going to be my transmembrane potential, which is a fancy way of saying the electrical difference across the membrane, and that's going to be measured in millivolts. So if we look here, we're going to start out at minus 70 millivolts. So this is my resting membrane potential. And threshold, remember we said, is minus 55. Now that varies. Some cells it's closer to minus 60. And so that's my target that I need to hit if I want my cell to fire. If I stimulate the cell and some sodium moves in, but I only get to about here, I haven't reached threshold, so I'm not going to fire. I'm just going to go back to resting. But if I get to here, that's when those sodium gates are going to open. Sodium is going to rush into my cell until I get to positive 30. So as sodium is rushing in, we said that is called depolarization. When I hit positive 30, remember my sodium is going to close and my potassium gates are going to open. Potassium is then going to rush out, and it's actually going to overshoot a little bit, and then it'll come back to resting. As potassium rushes out, we called that repolarization. And eventually I'm going to end back at minus 70 millivolts. But notice that I did overshoot a little bit here. So now I'm down at about minus 80 millivolts, which means I am hyperpolarized. So this is hyperpolarization. Now, neurons follow the all or none principle, meaning either they fire or they do not. You can't be a little bit pregnant. A gun doesn't partially fire, right? It is or it isn't. So once my neuron is in the middle of firing, it cannot fire again until it has completely reset. We call that the refractory period. So once I hit threshold here, until roughly here, this is my absolute refractory period. I absolutely cannot fire again while I'm in the middle of firing. It's kind of like asking a runner to run a 100-meter dash in the middle of running another 100-meter dash. You can't do it. You have to finish the first race before you can start the second one. Now, once I hit that minus 55, until I'm back to kind of this minus 70, remember I'm hyperpolarized. So this is my relative refractory period, meaning I just finished a race and I really don't want to run again right now, but if you kick me extra hard, I will. So if you give me this extra 10 millivolts, and so you're giving me a 25 millivolt jolt instead of a 15 millivolt jolt, then I can fire again. Now, to relate refractory period to something you might already be familiar with, this is a term that is used in other aspects of physiology and medicine. And one way you can think of it is that men have a refractory period. There's a certain period of time when they cannot perform again, so to speak, until the body can reset itself. So same idea here, my neuron cannot fire again until it is done with the current firing. So that's kind of a quick visual summary of what you learned about in the video and maybe adding a few more details to it. We'll start another video and talk about what happens when we actually get to that synapse.